So in the last lecture, we reviewed the basic structure of the central nervous system. We talked about how to navigate around the nervous system and what the different lobes of the cerebral cortex more or less do. As cognitive neuroscientists, we're interested in examining brain behavior relationships. So we observe a particular behavior and we want to know what brain areas play a role in that behavior and how they might contribute to that behavior. And so what we're going to look at in this lecture are methods, neuroscientific methods, that allow us to examine brain behavior relationships in both humans and animal models. And so when we're going through these methods, what you want to think about is, does the method allow for a causal versus correlational investigation of the link between behavior and brain function? So when I say causal, I mean that does the method allow us to conclude that the brain area of interest plays a causal role in the behavior we are studying? Or is the link just correlational? And the second thing you want to think about is the spatial and temporal resolution of the technique. So spatial resolution refers to the ability of the technique to resolve fine details in the brain. Think about a really high-powered telescope that you might use at some sort of astronomy research center. That high-powered telescope can resolve very fine details of Jupiter. On the other hand, if you go to Canadian Tire and buy a $50 telescope, you'll be able to see Jupiter, but you won't be able to see all the details of it with that telescope. So that's spatial resolution. Temporal resolution refers to the ability of the technique to detect changes in brain activity over time. So some techniques are actually better than others at sort of rapidly detecting that neural firing has changed in the brain. So you want to think about both of these things when we are going through these different neuroscientific techniques. And the question that we ask, so whether we're interested in a causal type question, showing that a brain area actually plays a role in a behavior versus a correlational type question, whether we're really interested in the timing of a neural process versus locating it in the brain, the question we ask dictates the method that we use. So when we talk about the brain, we tend to relate different brain structures to different behaviors. But we didn't always think that way. So early neurophysiologists believed in a concept known as equipotentiality and mass action. And equipotentiality simply states that if you lose one brain area, other brain areas can step in and take over in a way. Mass action is a related idea, and it simply says that if, let's say, 30% of your brain is removed, this results in a 30% deficit to all behavior. So under these two ideas, we shouldn't see that brain damage leads to specific deficits in specific behaviors. But of course, that's exactly what we do see. And the first person to really note this and document this was Pierre-Paul Broca, a French neurologist who studied a patient named Tan in the mid-1800s. Now, Tan suffered damage in his left hemisphere in the frontal lobe, and Tan was unable to produce fluid speech following this damage. You can see Tan's brain on the slide here. Tan could understand speech. He could understand language, but he wasn't able to produce speech. So we see damage to a specific brain area results in a very specific deficit. So this was sort of the first evidence that functions, cognitive functions, might be localized in the brain. Shortly after Broca reported the case of patient 10, Karl Wernicke, a German physician, reported the case of a patient who had damage in his left temporal lobe, and he had really poor comprehension of speech, but fluid production. So here we're seeing kind of like the opposite problem to Broca's patient. Damage in a different brain area results in poor comprehension, but still fluid production of speech. So if we put these two patients together, we actually have what we call a double dissociation. And this double dissociation, and I'll explain that term in the next slide, suggests that these two related cognitive functions, producing language, perceiving language, are separable in both the mind and the brain. And it provides fairly strong, what we would call neuropsychological evidence that these two behaviors are localized to specific brain areas. In this case, what we call now Broca's area and Wernicke's area. So what's a double dissociation? Well, double dissociations typically come from studying patients who have suffered brain damage and have various deficits in behavior. And once in a while, we find a case where we have two related aspects of a behavior, let's say producing speech and perceiving speech, and we find that damage to one brain area causes a deficit in one of those aspects. So let's say, in behavior A, and spares behavior B, or is damage to another brain area causes a deficit in behavior B, but spares behavior A. So we see the opposite pattern of interference. And so this is exactly what we see with Broca's patient and with Wernicke's patient. Right, in one case, we have a patient with damage in Broca's area, left frontal lobe. This patient has really poor production, but their perception of language is fine. In the other case, we have a patient with damage, again in the left hemisphere, but in the temporal lobe this time, and they have impaired perception, but spared production of language. So these two related behaviors doubly dissociate. And it's called a double dissociation because they're separable in both the mind and the brain. So it's providing some evidence that these two related behaviors are viewed as different in the mind. They're different processes, but they also rely on different brain areas. And in neuropsychology, and neuropsychology typically refers to studying patients who have brain damage and linking the part of their brain that's damaged to a deficit in behavior. In neuropsychology, the double dissociation is really, the most powerful way to show that a particular behavior or function is localized to a particular brain area. Now, there are many problems with studying patients. First of all, we have no control over the site of the damage, right? Strokes happen. They happen in some brain areas more than others, and we have no control over the extent of the damage. And so there's a lot of variability between patients in terms of what part of their brain is damaged. The other thing to consider is that there's some amount of neuroplasticity. So although the idea of equipotentiality in the strictest sense is wrong, if you lose a part of your brain, other brain areas can't just step in and totally take over that function, there is some amount of neuroplasticity. So if you suffer brain damage, other brain areas can step in and help out a little bit. And so we do see some recovery of function following brain damage due to neuroplasticity. But we now have a technique for inducing what we call a virtual lesion in the lab. And we can do that using something known as a non-invasive brain stimulation. In particular, transcranial magnetic stimulation, or TMS. And we call this a virtual lesion because what we're doing is we are applying a brief magnetic pulse to the head, and you can see in this image, this is what's called a TMS coil, and we simply run electric current through this coil very, very quickly, and this generates a magnetic pulse that travels through the skull into the outer layers of the brain. And what this pulse does is it disrupts neural firing in the stimulated brain area. And so single pulses of TMS can disrupt neural firing for 100 milliseconds or so. Repetitive pulses of TMS can actually cause the part of the brain that you're stimulating to be inhibited, so sort of turn down for up to 15 minutes. You can also turn up different parts of the brain. And so TMS allows us to basically induce virtual lesions in healthy human participants in the lab and see how this alteration in their brain impacts their behavior. And what's nice about TMS is that this is a causal method. We're doing something to the brain and we're seeing a change in behavior, right? So if I stimulate your primary visual cortex with TMS and you see flashes of light, which is exactly what would happen if I were to do that, I could conclude that, primary visual cortex is involved in light perception. So TMS is a really cool technique. There are some downsides that I'll talk about in a second. I'm going to show you a video filmed by the BBC in which the reporter receives TMS. I have some expertise in TMS. We're getting a TMS device here at Acadia. I've had TMS, 30 or 40 times with no ill effects that I am aware of, although perhaps my students might disagree. But in this video, you're going to see the reporter receiving TMS, and it's going to mess up his ability to sing in this case.
If we start at the back of the brain, we've switched off my visual cortex, so made me unable to see things. If we move a little bit forward in the brain, we've interfered with people's ability and my ability to see faces. A little bit further forward, we've interfered with the ability to perform actions. Start moving your finger.
It's quite odd.
It's not your finger anymore.
It's not my finger anymore.
We've interfered with the ability to compute with numbers.
6, 7, 8.
Okay.
Ooh, yeah. Yes.
We've interfered with the ability to speak.
London Bridge, London Bridge, London Bridge is falling down.
You think of something you can do, and we've basically interfered with it with this machine.
2, 3, 4, 5, 6, 7, 8, 9.
So what's the problem?
I don't know.
So as you saw in that video, the reporter was receiving TMS, and it was causing muscle twitches. So if we stimulate motor cortex, we can actually cause movements of your limbs. If we stimulate over Broca's area, we can induce speech arrest. If we stimulate over the right side of your brain, the frontal lobe, we can actually cause song arrest, which is what you were seeing in that video. If we stimulate primary visual cortex, we can make you see flashes of light, which are known as phosphenes. And again, what's nice about this method is it's kind of like we're studying patients who have had, brain damage, but the brain damage is temporary. And because we're physically doing something to the brain, it's a causal method. We are altering brain function and we're seeing a change in behavior. And so we can conclude with certainty that the brain area we're stimulating is involved in that behavior in some way. Now, there are some downsides to TMS. So with TMS, we can really only stimulate the outer layers of the brain. So subcortical structures like the basal ganglia, we can't stimulate that structure with TMS. We can only really go after the cerebral cortex and the cerebellum. TMS can be somewhat uncomfortable, I would say bordering on painful, depending on where you stimulate. But it is, really sort of cool technique for linking brain function to behavior causally. The other thing about TMS is that it has really high temporal resolution, right? So you're sending a pulse of magnetism into the brain, and that's almost having an immediate effect, an immediate change is occurring in the brain on the order of milliseconds. So the effects that we see are very rapid, which means this device is very high temporal resolution. Its spatial resolution is sort of so-so. We can target brain areas that are, about a centimeter in diameter or so. So it's not super precise. So let's talk about some correlational techniques, some techniques for correlating brain activity with behavior. So one of those techniques we can do with animal models, and you already saw a little bit of this in the last lecture. I showed you that video of Hubel and Wiesel's famous experiment where they're recording from primary visual cortex in cats. So in animals, we can implant electrodes directly into the brain and record from either single neurons, this is called single unit recording, or record from groups of neurons, which is called multi-unit recording. And this allows us to correlate brain activity with behavior. Now, this method has some benefits. So because we're recording directly from the brain, the temporal resolution is quite high, almost instantaneous, right? Neurons fire, we immediately record that firing. Spatial resolution is also incredibly good, right? It's arguably as good as it can get because we can record from single neurons in the brain. The obvious downside here is that we can only do this in animals, right? I can't recruit undergraduates to come into my lab and implant electrodes in their brain and record from single neurons. So we can only do this work in animals. In humans, in healthy human participants, the closest we can get is EEG, so electrocephalography, and we run these experiments here at Acadia. And in these studies, you have electrodes placed on your scalp, and what these electrodes do is they measure the firing of neurons through your scalp and through your skull. And you can see somebody with electrodes on their head in this image here. Now, a lot of EEG experiments involve the recording of what's called event-related potentials. This is also called an ERP, that's the acronym, event-related potential. And the idea here is that the EEG recording is time-locked to a particular event. So what does that mean? It means that you've got these electrodes on your head, you're doing some sort of task. Let's say it's, responding to a visual stimulus that appears on the screen. And you'll do this task over and over and over again. And the EEG signal from the brain area of interest, maybe the occipital lobe in this case, is averaged starting from the point where the stimulus appears on the screen. And if we do this, we actually see a very sort of stereotypical response. And this is the event-related potential. And there are different ERP responses for different cognitive behaviors. So the ERP response for visual processing might look a little bit different than, let's say, the ERP response for somatosensory processing. And so we can investigate, how these responses change. If you're attending to a stimulus, does this change the ERP response, for instance? So this tends to be how EEG is used. EEG has very good temporal resolution. So neurons fire in the brain, and these EEG electrodes can, pick up that firing, can detect it very quickly. It has very, very poor spatial resolution. And that's because the skull actually distorts the electric fields given off by these neurons firing. And so it's hard to say with certainty where activity is occurring in the brain when we record it using EEG. So EEG is great for questions that require, high temporal resolution. It's bad if our question is more about, localizing, pinning down a particular function to a particular brain area. A related paradigm is known as electrocorticography, or ECOG. And ECOG basically involves taking EEG electrodes and implanting them actually on top of the brain under the skull. Now, this doesn't happen very often. We typically only do this in patients who are awaiting surgery for epilepsy. So, epilepsy can be treated by removing the part of the brain where the seizure starts. There's basically, I don't have any expertise in this, but my understanding is that there's some sort of miswiring, and so the seizure starts in a particular brain area and spreads then throughout the brain. And this can be treated by removing that brain area, but the doctors don't know where that brain area is. They don't know where the miswiring is in the brain. And so what they do is they open up your skull and they implant this electrode array onto the surface of your brain. And then you just wait around until you have a seizure and the array detects, where the seizure began. And then the surgeons remove that part of your brain, and hopefully this cures or lessens your epilepsy. But while you're waiting, you can do psychology experiments. And so, research involving this technique happens throughout the US and Canada with these patients who are basically waiting to have a seizure. While they're waiting, they do psychology experiments. And ECOG has amazing temporal resolution like EEG, but it has much better spatial resolution because the electrode array is under the skull. And so with EEG, that skull normally distorts, the location of the electrical signal. With ECOG, we don't have to worry about that. Now, the downside, of course, is that there aren't a huge number of patients having this procedure at any one time. So your participant pool is very, very limited. So another popular technique that you may remember from intro psych is magnetic resonance imaging, or MRI. And we can do either structural MRI, so taking static pictures of the brain, that's what you see on the slide here, or functional MRI, where we're basically taking movies of brain activity. And MRI is based on this idea that if you're placed in a strong magnetic field, the hydrogen protons in your body will all line up. If we energize them, when they fall back down, they give off a signal. And the signal that they give off varies depending on the tissue these protons are embedded in. Right? So if they're embedded in white matter versus gray matter, the signal these protons give off when they fall back down into their unenergized state will be different. So the signal varies as a function of nearby tissue. And so some very smart physicists figured out, we can put people in a big magnet, their hydrogen protons will all line up. We can energize them briefly and detect the signal these protons give off when they fall back down to their unenergized state. And based on that signal, we can actually create an image of the brain that shows the different structures in the brain. And you can see that on the screen here. Very nice depiction of the cerebellum, corpus callosum, brainstem, and then you can see the sulci and gyri of the cerebral cortex. So very cool. So this is structural MRI. Functional MRI works on basically the same principle, but some other smart physicists figured out that the signal that these protons give off when they fall back down to their unenergized state also varies depending on whether there's oxygenated blood in the general area. Why does that matter? Well, when your brain is active, it requires more oxygen. And so what this means is that parts of your brain that are really active, so let's say you're doing a task that involves a lot of visual processing, which is what you're doing right now, or auditory processing, you're perceiving speech, there's oxygenated blood flowing to those brain areas. So in the case of visual processing, the occipital lobe. In the case of auditory processing, the temporal lobe. And so the signal given off by those hydrogen protons not only varies as a function of the tissue they're embedded in, but it also varies as a function of how much oxygen is nearby. More oxygen means more brain activity. And so some very smart physicists were able to figure out how to use this information to basically create movies of brain activity in relation to participants doing something in the MRI scanner. So you're put in an MRI scanner, and you can see an image of that here, and you're asked to do some sort of task. And while you do this task, the signals given off by protons in your brain as they fall from an energized to unenergized state is measured, and we can then work out what parts of your brain were active during that task. So this is a correlational method. We're correlating brain activity with behavior. So what that means is that if we see that a particular brain area is active during a behavior, we can't say with certainty that that brain area actually plays a role in that behavior. We can only say that it's associated with that behavior. fMRI has better spatial resolution than EEG. So spatial resolution is actually, pretty good, on the order of centimeters to millimeters. Its temporal resolution is quite poor, and that's because it takes some time for oxygenated blood to flow to active brain areas. So when we see a change in the MRI signal, that actually reflects something that's happened some time ago, because it's taken some time for oxygenated blood to flow to that active brain area. So what's cool about a lot of fMRI studies is that they use this attraction method first pioneered by Donders in the 1800s. And so if you recall, Donders showed that, if we want to study decision-making by looking at reaction times, we need a decision-making task, and then we need another task that involves everything the decision-making task had but for that decision. And the difference in reaction time reflects the mental effort associated with the decision-making process. fMRI studies use the same idea. So you'll be put into a scanner and, maybe the researcher is interested in, let's say, face perception. And so you're shown faces and your brain activity is recorded. This needs to be compared to something. So then you'll be shown, objects or scrambled faces and your brain activity is recorded. And the brain activity in the first case is subtracted from the brain activity in the second case. And if you've designed your study correctly, the remaining activity should reflect the behavior or psychological process that you're interested in examining. So this is some data from a very famous study that looked at a part of the brain that seems to be involved in face perception. It's called the fusiform face area in the temporal lobe. And so what they did in the study was they showed people pictures of faces, and they showed them pictures of objects. And they simply compared the fMRI signal during face perception to the fMRI signal during object perception. And they're looking at the specific brain area in the temporal lobe. And what you can see is that the signal actually goes up during face perception and then drops during object perception. Goes up during face perception and then drops during object perception. And so it's, really nice correlational evidence linking this brain area to a specific cognitive process, in this case, perceiving faces, which is something that we do every day and is arguably, very important to us. So based on everything I've said so far, I've probably given you this, picture that every single aspect of our cognition and our behavior can be linked to a different brain area. Well, that is far from true, and actually real-world cognition is achieved through interconnected neural networks. And any one cognitive or behavioral process probably relies on, dozens of brain areas. And just to illustrate that, this is a model of language processing. And I don't expect you to, learn this model or even understand what's going on here. The point of the image is just to show you how complicated it is. And we actually think that, during language production, during language perception, we're relying on neural networks that break off into two distinct streams, a dorsal stream and a ventral stream. We will talk about dorsal versus ventral streams in the context of visual perception. And so again, the idea here is that visual perception, in this case, perceiving objects, relies on one particular neural network, and another aspect of visual perception, perceiving object motion, relies on a different neural network. But the take-home message is that real-world cognition is achieved through interconnected neural networks, so different brain areas sort of working together. Another idea that comes up in cognitive neuroscience, and this is based on functional magnetic resonance imaging, is known as functional connectivity. This stuff is really cool. It involves putting people into an fMRI scanner and having them do nothing and just lie there. And you measure their brain activity while they are resting. This is called a resting state scan. And what was discovered, I guess it's about 10 to 15 years ago now, is that when you're lying there at rest, first of all, there's no, when you're lying there at rest, it's not like all the neurons in your brain just shut off and stop firing, right? They're still firing. There's a baseline level of activity that's happening at all times. But when you're lying there at rest, different parts of your brain fire in synchrony. So the activity of different parts of the brain is actually correlated. And we call this a functional connection. So two areas that have highly related activity, motor cortex and somatosensory cortex. So when you're lying there at rest in an fMRI scanner, we can see that the activity in your motor cortex is highly correlated with the activity in your somatosensory cortex. And that kind of makes sense because motor movements and touch perception, proprioception kind of go, pardon the pun, hand in hand, right? So you kind of need one to do the other and vice versa. And so again, the idea here with functional connectivity is that if you're lying there in the scanner and we see a high correlation between two brain areas, we can say that they're functionally connected. Now, a functional connection doesn't mean that those brain areas are necessarily directly connected, physically connected. It suggests that they are, but we don't actually have evidence for that. This is correlational data. So why is this interesting? We can examine how these connections change with, for instance, learning. So if you do, some sort of motor learning task, let's say you learn how to, swing a baseball bat or swing a golf club, do we see an increase in the functional connectivity between motor cortex and somatosensory cortex? And this would suggest that these two brain areas are associated with motor learning. And, in this case, I would assume that they would be. So this method allows us to show how brain areas, might work together in neural networks to contribute to a behavior. I'd like to end the lecture with this image, and I borrowed this image from Professor Matthew Rushworth's Introductory Psychology Lectures. Matthew is a professor in the psychology department at Oxford, and I think this image nicely sort of shows the relationship between the different techniques we've talked about, whether they're causal versus correlational, and their spatial and temporal resolutions. So, causality relates to red versus purple. So the purple techniques are correlational in nature. We didn't talk about positron emission tomography in the lecture, but it's a similar idea to fMRI. On the x-axis here, we have the temporal resolution of the method. On the y-axis here, we have the spatial resolution of the method. ERPs, electrocephalography, as we discussed, has very high temporal resolution, but very poor spatial resolution, right? So we can't pin down exactly where the neural firing is coming from. We can detect it very quickly, but we're seeing it sort of on the level of the entire brain. On the other hand, if we record from individual neurons, right, we have very high spatial resolution and very high temporal resolution. And so I think this image nicely shows how some of the techniques that we've discussed in this lecture differ in terms of spatial and temporal resolution, and it also nicely shows whether they're causal, so the red outlined areas, versus correlational. So we've come to the lecture review. Question 1. What does a neurological double dissociation typically show? Question 1. What does a neurological double dissociation typically show? So the idea here is that related mental processes are separate in both the mind and the brain. So the example in this lecture is producing language versus perceiving language. Two related processes, but we've found patients that have damage in different brain areas, and one shows a deficit in perceiving language, but their production is fine, and the other shows the opposite pattern of behavior. Double dissociations allow us to say with some level of certainty that these two processes are separable in both the mind and the brain. Does TMS allow us to test whether a cortical area plays a causal role in a behavior? So question 2, does TMS allow us to test whether a cortical area plays a causal role in a behavior? The answer, of course, is yes. What's nice about TMS is that we're doing something to the brain. We're inducing what's called a virtual lesion. And if we see a change in behavior, we can say with, some amount of certainty that that brain area is contributing to the behavior that's altered. Question 3, true or false? EEG has high spatial resolution but low temporal resolution. Question 3, true or false? EEG has high spatial resolution but low temporal resolution. This is, of course, false. EEG shows the opposite pattern, so great temporal resolution but poor spatial resolution. ECOG has better spatial resolution and just as good temporal resolution. But as we discussed in the lecture, ECOG involves putting basically EEG electrodes under the skull on top of the brain, and we can only do that in a subset of patients who are waiting for surgery for epilepsy.